Alrighty, here we go. Let me click this. Whoopsies. Let's try this and I'll minimize this. Alright, so hopefully everyone can hear me and see me okay. I will be recording this and I'll upload it up to the portal for you guys as well so that you guys can watch it ahead of time. Sorry, after the stream is all done. I won't be keeping it up for that long. I'll probably keep it up for about a week or so, and then I'll pull it down. So today, essentially, what we're going to be doing is we're going to be finishing up on biological membranes. So this is going to tie in nicely with what we were doing last week, looking at lipids, and sort of try and wrap up the course and looking at our final section of proteins, because we've covered fibrous proteins and we've covered globular proteins. We're not going to go as in depth with biological membrane, membranous proteins, but we're going to be talking a lot more about the biochemistry of the lipid bilayer and why it is so sort of crucial for human function. All right, so we can see straight away much fewer learning objectives here. So big important thing here is we're going to be looking at the fluid mosaic model, and importantly, we're going to be looking at the factors that affect membrane fluidity. So what I mean by that is with our lecture last week when we were looking at lipids, a big thing I mentioned is that the physical properties of lipids can change depending on that lipid's makeup, depending on whether it is a saturated fatty acid or a monounsaturated or polyunsaturated fatty acid. And a good example of that was when we were looking at like lard, or animal fat sort of thing, compared to, say, olive oil. So if you've got both of those at room temperature, one of them is a solid, one of them is a liquid. And that's down to the number of carbons in that fatty acid chain, and down to that saturation. Now, this same thing in terms of whether the fatty acid is going to be a liquid or a solid at room temperature or physiological temperature depends very much so on the properties of those lipids, and our cell membranes are no exception. So that's what we're going to be talking about with our fluid mosaic model. We're going to be looking at the methods in which lipids can move in the lipid bilayer, because it is not a fixed structure. Things are constantly moving around. Very important for cell function. Sorry, I'm going to try and be muting my microphone wherever I can. I'm still getting over a head cold. We're going to be looking at the three types of membrane proteins with respect to structure, isolation, and function, so whereabouts we sort of typically see them and their functions there. And we're going to be looking at hydropathy plots. So on that, now we're going to be looking at membrane components, looking at lipids and cholesterol. So when we're looking at lipids, when we're looking at the membrane, a lipid bilayer, what is their primary role? So without it, essentially, we would not be able to exist, nowhere near. So it controls, it's basically a gating off the movement of substances inside and out of a cell. Now, this can seem very straightforward, very common sense. You want to keep some things out, you want to keep some things in. But it's much more important. It goes much deeper than that, because we need to be very careful to control the environment, because there are a lot of things where a lot of environmental conditions or conditions within the cell for a chemical reaction to occur needs to be under a very specific set of conditions, and we might need different conditions for different parts of the cell. And a perfect example of that is looking at our organelles and things like lysosomes. So we don't want, those those enzymes that are responsible for, breaking down, various molecules within the cell to be just exposed everywhere. We want to contain them. And likewise with our cells, we want to keep those nice and contained as well. We don't want them leaking out. It also contains receptors for signaling molecules that turn on or off processes within the cell. So essentially, it's like having an antenna. It's like a receptor there to control, communications from outside the cell and inside the cell. Here we go, so that way we can see it properly. The third point contains a fuzzy coat of glycoproteins and glycolipids, which are crucial to cell-cell attachment and cell recognition. So think of it like Velcro. These glycoproteins, glycolipids, for instance, can work as like little hooks and can hook one cell onto another cell. Again, very important for cell-cell recognition, like in the immune system. Or being able to recognize, like, say, apoptosis, so regulated cell death, things like that. That is essentially communicated by receptors that are presented on the cell membrane. Excuse me. So again, just a bit of a recap on what we did last week. So we looked at the cell membrane and we looked at some of the primary constituents. So last week we looked at glycerophospholipids and we looked at sphingolipids. The week before we looked at carbohydrates, and of course, for a majority of this course, we have been looking extensively at proteins. We'll be looking a little bit more at cholesterol today with respect to the function of the lipid bilayer. Not going to touch too much on glycolipids and glycoproteins. As I said, I sort of touched on that a little bit when we did look at saccharides, so I'm not going to carry that too much further. Now, big thing to keep in mind, big thing to remember here is that biological membranes are impermeable to most polar or charged solutes. So essentially for those, so polar solutes or, solutes that have a charge or even sort of larger molecules require transporters to move through. And that's one of the main membrane-bound proteins. However, there are a lot of non-polar compounds, like steroidal-based hormones, that can freely move straight through the lipid bilayer. So again, just a bit of a recap. Down at the bottom, we've got our sphingolipids, we've got our sphingosine backbone with our fatty acid down here. And just to compare that to a triacylglyceride, so we've got that, so we've got our phospholipid here, we've got a polar phosphate group, polar head, with our two different fatty acid tails. And of course, we've got the structure for cholesterol. Now, I don't expect you to be able to draw cholesterol or have that sort of memorized. However, I do expect you to recognize it. So again, big giveaway here is 6665, 6665 carbon ring. So when we see 3 6s and a 5 carbon ring joined together, it is indicative of a steroid group. Now we do have this hydroxyl group down at the very bottom here, giving this molecule a bit of polarity, so it's a sterol, and we can see sort of a longer carbon chain at the top here. So these are the 3 things I sort of use to recognize cholesterol. Now, again, plasma membrane, a large focus of today. So it's a basic structural unit of the plasma membrane is the phospholipid bilayer. So the big important thing is that it has this hydrophilic head. So if we just go back a slide, this is this phosphate group here. Then we have these hydrophobic fatty acid tails. So these hydrophilic heads are obviously facing outwards and inwards towards the extra and intracellular surfaces. And then we've got these hydrophobic tails that are sort of adhering together via these hydrophobic interactions. Now, they are a somewhat fluid and flexible structure. This is very important, okay, because we as an organism, we as a creature are quite fluid and quite flexible, okay? We are able to move, to bend, to, and some people more than other, squish, okay? So the big thing here is that it's good to be fluidic, it's good to be able to control that, but that's sort of the key point. We need to be able to control how fluidic we are. Because fluidity, especially when we're looking at lipids, depending on temperature, on lipid composition, and this is something I'm going to talk about more as we move forward, is looking at the cholesterol content. Because the majority of cholesterol, where it is found, is in our lipid bilayer. Now, the formation of a biological membrane. Again, this was discussed last week, so I'm not going to spend too much time on it, but essentially it is the secondary attractive and repulsive forces. As I've mentioned, we have these hydrophilic polar heads with these hydrophobic fatty acid tails. Sorry, let me bring that back like this. Now, what this means is that the formation of this lipid bilayer is due to the driving force of our secondary attractive. Okay, these polar head groups, these hydrophilic polar heads, are attracted to water, so they are wanting to move towards that water. Whereas these hydrophobic fatty acid tails are having those secondary repulsive forces. They are saying, no, stay away. I do not want to be anywhere near that water. So what is going to happen is that the lipid bilayer is going to be organizing itself so that these polar heads are facing the extra or intracellular fluid, again, because that's primarily water. So let me just move that down a little bit. There we go. And whilst at the same time, those hydrophobic fatty acid tails are having that repulsive force, wanting to move away from the water, while simultaneously having that secondary attractive force with each other, because they are both hydrophobic. Now, when I say both, I mean obviously the fatty acid tails of, let's say, a neighboring phospholipid here. So like this fatty acid tail and this fatty acid tail will be having secondary attractive forces in the form of hydrophobic attraction. Excuse me. So, glycerophospholipids, sphingolipids, and sterols virtually insoluble in water, so they will try and move away from that. And again, this is what we see with these vesicles. It's going to curl itself around in a ball. It's not going to be like this perfectly cut rectangle here, because all this side here would be exposed to the water that's found inside of our body. Same with over here, so it will dome itself in a circular shape. Okay, now, as nice as it is to go, yes, this is our membrane, and that's all you need to know, job done. Unfortunately, again, it is a lot more complicated than that. So in the real world, biological membranes are composed of different types of lipids and various compositions. This changes not only by cell type, so if we were to compare, say, skin cells versus skeletal muscle versus smooth muscle versus, all these different types of cells that are found in the body, what we would find is that those plasma membranes will have a different plasma membrane composition. Now, not only that, but we also note that there are different lipid compositions for organelles inside of those cells. They have a different lipid composition. And not only that, but whether it is the outer or innermost layer of our cell membrane, they can have different lipid compositions as well. They're not the same. So it does get very, very complicated. And I don't expect you to know, the specific lipid, compositions of various cells. I just wanted you to be aware that, they're not perfectly symmetrical, that they can be different. Okay. So this is essentially what I was explaining before. So membrane compositions can change with different organelles as well as cell types and the membrane layer, whether it is the outer or the innermost layer. So we can see here, for instance, we've got phosphatidylcholine. We don't see all that much of it in the inner layer, but it's much more prevalent in the outermost layer. And same with sphingomyelin, which we know we can see those in myelinated cells, so looking at our nerve and brain tissue. Whereas if we look at phosphatidylserine, much more of the inner lipid bilayer is comprised of phosphatidylserine. Again, not important to know the details. It is just important for you to just have that awareness that the composition does change. Because again, structure equals function. So if we do want different cell membranes as a part of different cells, part of different tissues, to have a different function, to do different things, then they must have a different structure. Okay, now the fluid mosaic model. What does this describe? Okay, it's quite an eloquent title. It's quite an eloquent description. But essentially, what it describes is it's like a floating ocean of lipids. Imagine that you sort of got like a swimming pool, and you sort of threw a heap of like floating balls in there, and you would see sort of those balls sort of moving and floating around on top of the water. That's essentially what this sort of looks like, and that's how I want you to sort of envisage it in your mind. Now these lipids here are in a constant state of motion. Again, it's not just rigid, it's not just sitting still, because if it was perfectly rigid, it wouldn't be that flexible, it wouldn't be able to move around. And us being able to have a clear fluidity and motion within the cell membrane is incredibly important. Incredibly important. So, how do these lipids, how do they move? Okay, so again, as I was saying before, membranes are not static. Okay, they are constantly moving around. Now they're in constant motion. The layers move over each other based upon the percentage of unsaturated fatty acids. So more unsaturated equals faster movement. Why is that? Again, what we need to remember is that the more unsaturated our fatty acids are, they have a kink in the chain. They're not perfectly straight. They'll have that kink. Now, if we've got our unsaturated fatty acids that don't have that kink, they can stack a lot closer to each other, have more of those secondary attractive forces between them, meaning it takes much more energy to break them apart because they have much more non-covalent interactions holding them together. What that means is, again, it takes more energy to peel them apart, meaning that they are most likely to be solid at room temperature. However, the more unsaturated it is, it has that kink, it means it's harder for those fatty acids to sort of stack together, meaning that there's fewer of those non-covalent interactions occurring, meaning that it requires less energy to break them apart, which in the case of looking at our membranes, meaning is much more fluidic. So, this next part I want to talk about is this lateral diffusion. So the movement is parallel to the membrane surface. What that essentially means is, if you have a particular, if we look at, say, just this fatty acid on the, facing outside the cell, it can move left to right and forward and back. So if I look at this picture, this might be a bit of a better example here. So if I just right where my mouse is, if we look at that, say, phospholipid, it can just be moving around like a person in a crowd. Okay, it can move forward, back, bump around, and they'll just sort of shuffle and move around each other. And that's quite normal, and it does that a lot. And this has been seen multiple times in studies and experiments. If you add a fluorescent marker sort of from two different cells or in two different parts of the cell, they will eventually move and mix together. However, the big thing here is that although they can, it takes a very, very, very long time, it's very difficult, is it can flip-flop. Now what I mean by flip-flop is that if we come back to this example I was talking about over here, how this fatty acid, or this phospholipid I should say rather, is facing the outside of the cell, it can flip, it can turn upside down, which means this hydrophilic polar head is now pointing towards the inside of the cell. Now it's very difficult for this phospholipid to be able to do this because obviously this polar head is hydrophilic, these tails are hydrophobic, so it doesn't want to flip around to the other side. It can do it, but again, very, very, very slow, and it's not something that happens frequently. However, we can have enzymes that can flip-flop these particular phospholipids. And it's called flipase, which is just wonderful. I love the name of that. So what it does is, again, it'll take that phospholipid and will flip it upside down. And this is very, very important, especially with like glycolipids. So these lipids that have sort of a saccharide attached to it. So like, again, presenting a receptor to the cell surface, stuff like that. It can take advantage of these mechanisms to sort of like a flag, raise it, and then sort of hide it again. Pardon me. So, important things to take away from this is that these fatty acids will typically move side to side on that same layer. They can flip-flop, so they can go from facing outwards to facing inwards and vice versa. However, typically it will require the assistance of an enzyme to be able to do that. Now, let me just swap to this scene. Here we go. Just so you guys can see the whole slide. So, membrane degree of fluidity. So, depending on their composition, so the composition of the lipid bilayer, and of course the temperature in which it is exposed, it can be in either a more like paracrystalline state, so sort of a quite solid, quite rigid state, or it can be in a very, very flexible and more fluidic state. So this is what I was describing earlier when I was discussing the degree of saturation of our fatty acids that are seen with our lipid bilayer. So we can see with our lipid bilayer when it's in this paracrystalline state is that these sort of fatty acid tails, very rigid. We're seeing very close together with surrounding phospholipids. Lots of these secondary non-covalent interactions going on here, supporting it, very rigid. However, once we heat things up, we change things up a little bit. Let's say we've got some more unsaturated fatty acids that have got more kinks in the chain. We're seeing a lot more disruption. We're seeing a significant increase in this fluidity in that lipid bilayer. It's not as sort of rigid and sort of stiff as this one. Now, TM is basically just indicative of that sort of transitional temperature. Transitional temperature of when it's sort of much more of this sort of solid and rigid like structure versus becoming much more fluidic. Now, when it's in this solid phase, the individual molecules don't sort of move and shuffle about. And that's just because of how rigid it is. Whereas when it's in this more fluidic state, they can move and jostle around. Now again, I'm going to talk about that a little bit more as we move forward and explain that. But again, I just wanted to sort of link that to what we did last week when we were talking about lipids. Whoops. So, the fluidity of these membranes, again, it all comes down to structure and function. And there are some lipids, there are some sort of our membranes that we want to be very, very fluidic, very, very flexible. And there are others that we want to be much more rigid, much more fixed. Another problem too is that as we sort of showed over here, this is a very, very steep curve, and especially if temperature is a factor, we really don't want to have such a sharp change between whether it is much more sort of rigid or much more fluidic based on a small change in temperature. You know, you sort of don't want to walk around in the snow, like, you know, you just say take your sock off and put your foot straight in the snow, and then suddenly, boom, your foot goes super rigid and you're unable to move it. Versus, you know, or just you be exposed to like a hot summer day and then suddenly, you start turning into like rubber sort of thing. We don't want that. We want to have a greater degree of control over the fluidity of our membrane, and we're able to do this by controlling this transition temperature, the TM. And we do this by controlling the degree of interactions between lipids. So the transition temperature is dependent on the length of the fatty acids in the membrane lipid and the degree of this cis unsaturation. Again, everything I was repeating last week in class. But the big sort of takeaway message here is the use of cholesterol. So what cholesterol does is allow proper control over this membrane fluidity. So by putting cholesterol in here, it weakens and sort of controls the degree of non-covalent interactions that are occurring between these fatty acid tails. And what this has is a sort of significant change on our curve. So if we have a look with no cholesterol, it's a really sort of sharp sigmoidal curve here in which we see a very, very sharp change in the fluidity of our cell membrane based upon a change in the temperature. However, when we add that cholesterol, it's a much more gradual change. It basically means that by causing this interruption to these non-covalent interactions between the fatty acid tails and sort of reducing the overall number of those, meaning that we can greater control that fluidity and not be so sort of critically reliant on temperature. So just to quickly recap on what we've been talking about sort of thus far. Again, the longer the fatty acid tail, the lower the fluidity, okay? The more rigid our overall structure will be. And that is due to, again, how nicely these fatty acid tails line up and the non-covalent interactions that are occurring between these fatty acid tails. Now the saturation of these fatty acid tails it will decrease that fluidity for the exact same reasons that I said below with the longer fatty acid tails. Okay, the longer it is, the more of those non-covalent interactions that can occur between them. The more saturated they are, they're not going to have these kinks and bends and sort of be all over the place. They're going to be nice and structured, plenty of interactions between them. Now, cholesterol can increase or decrease the fluidity depending on that transitional temperature. So cholesterol broadens it, okay, because our cells, our body has the ability to control how much cholesterol we are putting in these cells. And this is very important because it means that our body then has the ability to determine and easily control the fluidity of our cell membranes without relying on body temperature or environmental temperature for that instance. Okay. So, lipid rafts. These things are pretty cool. So, a lipid raft. Thinking back, if we go back to this slide here when I was explaining the fluid mosaic model, and if I was to say, it's like an ocean. Like, we have, if you're a boat in the ocean. You're a boat in the ocean and, you've got the waves moving up and down. You're a boat, you're sort of going to move up and down along those waves as well. That fluid mosaic model is sort of embodying that. So when I move on and sort of talking about these lipid rafts, I want you to be quite literal. I want you to really be thinking about a raft, or like our boat, that is that's on the ocean. That's sort of riding along with those waves. And essentially what it is that these lipid rafts are just more dense sections of these lipids that have series of, say, proteins or glycoproteins or glycolipids sort of adhered to them. And they can sort of move as a cluster around that cell surface. So instead of just having sort of one lipid moving side to side, like I was sort of describing over here, sort of moving around by itself, we can instead have what are called these lipid rafts, and it's a cluster of them that move around as a big cluster. And this is particularly important because we can have a lot of these sort of like anchored proteins that are anchored into this lipid, and when we, say, wish to present particular receptors for cell-cell signaling or adhesion, stuff like that, we can sort of present that lipid raft so they move together as a functional unit. Pardon me. So, they contain clusters of glycosphingolipids with longer than usual tails. So again, they want those longer tails to cluster together for them to be much more rigid and sort of hold together and be able to move around as a cluster around and sort of through the other phospholipids in the cell membrane. They are more ordered, they do contain more cholesterol, and they allow the segregation of proteins in the membrane, which is important, and it's believed to play a significant role in sort of cell-cell signaling. Seems a bit pointless to do a quiz when we're doing it online, but that's all right, we'll move on. Let me change it. Whoopsies, here we go. Now, this is sort of touching a little bit more again on the asymmetry of the plasma membranes. And I should probably move this a little bit earlier in the PowerPoint slides. But essentially all this is saying is just giving a bit of an example. So for instance, when we've got blood clotting, blood coagulation, it's only when phosphatidylserine that in that plasma membrane that moves to the outer leaf that blood clotting can be triggered. Again, not overly important that you do memorize all of that. It's just also just another example just to explain to you guys that asymmetric nature of our proteins. Of our membranes. What have we got here? Yeah, I've already explained all of that. Whoops, whoa dear, what's happened here? Okay, so again, just some key takeaway points from this. Looking at the biological membrane and the composition of that membrane is very important to the overall cell structure and function. It changes vastly depending on the type of cell because, again, each cell has a very different role within the human body, what it actually does. So, as such, it's going to have a different membranous composition. So, the membrane structure, it's best emphasized and described by this fluid mosaic model as in not being this super rigid and blocky like structure. It's able to move and flow. And we have these cool lipid rafts in which we can move sort of clusters of proteins and present proteins for cell-cell signaling and other various functions. So lateral movement is seen quite frequently within the lipid bilayer. It is also extremely rapid, but what we do not see is that flip-flopping. That does require a sort of that flipase, that enzyme, to be able to adequately flip it through. It can occur naturally, but again, much more unlikely, and it's much more sort of thermodynamically unstable. The motion of the membrane is or that fluidity of the membrane is determined by the temperature and the composition of the membrane, as I've explained before, and cholesterol plays a very crucial and central part in terms of that transitional temperature and allowing us to have a much broader range of fluidity without like such a sharp change based on a tiny movement in temperature. Now, so we've discussed the membrane, now the lipid bilayer, and we've looked at sort of the role of lipids in that. Let me change back to this one. So now let's have a look at some membrane proteins. So there are three main types of membrane proteins. We have our peripheral proteins, we have our integral proteins, and we have our lipid-anchored proteins. For the purposes of this course, so I will be talking about lipid-anchored proteins a little bit. However, it's peripheral and integral proteins. That's what I'm going to be focusing on a lot more. Pardon me. So the functions of these proteins do vary greatly, and again, it really does come back to that type of cell and what its overall function is doing. What is it doing? So they can be used as receptors, detecting changes from outside or inside the cell. We see the integral and peripheral proteins a lot with like hormones and neurotransmitters, which you guys will learn much more specific examples of those in your later studies. We've also got channels, gates, pumps. So channels and pumps, things moving into the cell, things moving out of the cell. How does it do that? Things like pores. You know, they require especially hydrophilic substances require a means to be able to move into and out of the cell. And it does that by us creating a pump or a hole inside of the lipid bilayer, which is crucial, and one of the many functions of integral proteins. And of course, enzymes. ATPase is a good example of that. That's actually embedded through a membrane. Again, it's not important that you know that. You'll learn that a lot more in metabolism. Now, starting off, let's look at peripheral proteins. So peripheral proteins are loosely associated with the hydrophilic heads found on the cell membrane, meaning that they sort of skirt the top or the bottom here. So they sort of shimmy along the top or shimmy along the bottom. What this means is, this is very important, is that it is simply held there by non-covalent interactions. Meaning that essentially these peripheral proteins can be quite easily disrupted, or they can quite easily be removed, especially in sort of a laboratory environment. Integral proteins, however, are a bit of a different story. So, integral proteins, what they do is they will essentially pierce through our lipid bilayer. Generally, they are asymmetrical. Not really important. Again, not a real focal point for you guys. A big one here is that it's very tightly associated with that lipid bilayer, with that lipid membrane. And it will be comprised of hydrophobic and hydrophilic sections within that integral protein. And the reason for that is it's sort of skewering, it's piercing through that lipid bilayer, which means there are sections, say up the top here, that will need to be sort of hydrophilic because it's sort of interacting with either a aqueous-based environment outside or these hydrophilic polar heads. Whereas this part down here that is actually piercing through the lipid bilayer, it has to be more hydrophobic because it's interacting with these non-polar fatty acid tails. Now, the only real way to remove these integral proteins is to essentially just destroy and break apart the membrane around it by using detergent. And this can pose a bit of an issue because detergent can also denature our proteins. So it can be a bit tricky to try and isolate an integral protein. Sorry for all the coughing, guys. So, integral proteins can exist in a variety of different structures, and this makes sense because they have a variety of different functions. They can do various different things. However, inside the lipid bilayer is predominantly an alpha helix. Okay, it's predominantly that sort of alpha helical spiral that's piercing through. And we can have multiple alpha helical structures, which again, depending on that integral protein. So for instance, if we have multiple alpha helices, they can sort of group together and form a pore, causing an opening in the lipid bilayer. So, the structure and functions of membranous proteins has lagged behind research of other proteins. So, for instance, globular proteins like hemoglobin, myoglobin that we were looking at a few weeks ago. Why is that? Again, what I was alluding to before is because they are so difficult to remove. And that the process of removing the protein from the lipid using that detergent, removing the lipid bilayer there, often involves destroying the protein as well. So there are some examples of integral proteins here. So looking at selectin and integrin here, I'm going to talk about those a little bit more later on. Again, looking at transporters, moving things into and out of the cell, receptor enzymes, so the binding of a signaling molecule, so say a water-soluble hormone, binding that to that integral protein, and what that can cause or what can happen there is that this will trigger a downstream effect and will activate, say, a G protein coupled receptor, causing a signaling cascade as a secondary hormone system. You guys don't need to know any of that. That's just one example of how it's used. Okay, now this is, we're taking a bit of a trip to the past here. We're going all the way back to, I think it was like week 3 or week 4 when we were first learning about these alpha helical structures. Now, the important thing here, and the reason why it is an alpha helical structure for these integral proteins, is that they are really, really stable. And that's really, really important because remember with our alpha helical structure, so if it's a so it's a right-handed loop, so it's going to be moving around like a spring. Those R groups of the amino acids are facing outwards. What this means is that those R groups, if they are hydrophobic, can interact on the outside of our alpha helix. And in the case of an integral protein, as it's sort of going through, as it's going through this lipid bilayer, the helical structure that's passing through like this needs to interact with these hydrophobic fatty acid tails. And that's exactly what we see here. So the R groups are facing outwards. This means that the R groups in the alpha helix in integral proteins must be hydrophobic. So that gives us something to look for. So integral proteins need around about 20 hydrophobic amino acid residues in a row. And that's important because we can't sort of have, hydrophobic, 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 hydrophilic, hydrophilic, hydrophobic, hydrophobic. Because there's going to be that disruption there. It's going to be repelled away from those fatty acid tails and it's not going to be stable, so we won't be able to do it. So, we know that our integral proteins are comprised, especially this section of the integral protein that's passing through that lipid bilayer, it is an alpha helical structure. Now, we know that with an alpha helical structure, those R groups are facing outwards, which means we need to look for around about 20 hydrophobic amino acids in a row if we are looking at the protein sequence. So how do we do this? There's got to be an easy way, and there is. Hydropathy plots. Now, just to be clear here, I will not expect you to draw a hydropathy plot. I will not expect you to, graph one based upon sort of giving you data in a table and giving you a sequence. What I do expect is if I was to say give you a hydropathy plot like this, that you would be able to successfully read and interpret it. So, hydropathy plots, what do they do? It is essentially plotting each amino acid versus how hydrophobic they are. So, hydrophilic amino acids that are, again, very easy to dissolve in water, they like water, it will have a negative transfer of free energy, whereas hydrophobic will have a high positive transfer. It's thermodynamically unstable. It really doesn't want to go near water. So what we do is we take our protein sequence and we measure the amino acids and look at that free energy change. And it will give us this lovely little graph. And we can see here, we have these big clusters. And what that translates to is the number of alpha helical chains that are piercing through the plasma membrane. So let me just make myself disappear. Okay, so for instance, if we were to look at this first graph up at the top left corner over here, if we look at this hydropathy plot, we can see this sort of, oh yeah, we've got a few here that are hydrophobic, and then, oh, a huge spike that's really hydrophilic, and then, yeah, a little bit hydrophobic, no real trends, and then boom, we see this sort of big sort of mountain. We have Mount Hydropathy over here. And what this tells me is like, okay, we have, at least 20 amino acids that are all hydrophobic. And that tells me that it is likely that that section of our protein is going to be the alpha helical structure that will pierce through the plasma membrane. So, for instance, the protein glycoporin only has one alpha helical chain that's in that integral protein that's piercing straight through. Looking at our second protein, however, we can see that it has many more of these sort of frequent sort of spikes. So we can see we've got 1, 2, 3, 4, 5, 6, 7. And what that translates to is that with this integral protein, that there are 7 sections of that protein that are piercing or going straight through the plasma membrane. So this, I'm actually not 100% sure what this protein does. It could be a pore or something like that that's causing to cluster together, like so. So again, just to recap, I'm not expecting you to be able to generate these graphs or anything like that, but I would expect you to be able to read it, like I've done for you just then. So I've already sort of explained this, that we can form these beta barrels and form these pores within a plasma membrane to facilitate movement of substances in and out of a cell. Now, lipid anchors. Look, again, I'm not going to be spending a whole lot of time on this, but essentially they contain a covalently linked lipid molecule which will attach itself to the membrane, meaning that because it is covalently linked, it's incredibly difficult to actually remove that lipid anchor. Excuse me. So the lipid part can then become more part of the membrane. It's sort of anchored on there. It's held in tight and a lot firmer. And these GPI anchored, so this glycosylated phosphatidyl GPI, I'm going to call it GPI, are found only on the outer face of the membrane, so they can be quite selective. Again, not a huge focus for this section. Now, looking at membrane proteins within our red blood cells. So just a couple of examples here, because again, we're focused on red blood cells quite a lot. Red blood cells are incredibly well studied, and they are one of the simplest. So well studied, nice and simple means fantastic teaching example. So that's what I'm going to be using. So, as we can see here from this SDS page gel, so what this is essentially separation of sort of proteins that have been removed from a erythrocyte, so remember a red blood cell, based upon its overall molecular weight, so the size of that protein. We're going to be looking at three main proteins: alpha spectrin, beta spectrin, and glycoporin. So, glycoporin, interestingly enough, was one of the first transmembrane proteins that was sort of studied and isolated. Now, glycoporin is actually manipulated and used by malaria, the parasite, in order to gain entry within red blood cells. It is very, very hydrophilic on the obviously on these outer and innermost edges, and it allows the red blood cells to be able to sort of move smoothly and sort of to not cluster and sort of get caught on various things within our blood vessels. However, this is still something that is currently under research. So, spectrin. What does spectrin do? So, spectrin is a cytoskeletal protein that's non-covalently associated with the cytosolic side of the red blood cell membrane. So, basically, it's attached non-covalently to the inside of our red blood cells' lipid bilayer. And what this does is, I want you to think of it as like springs in a mattress, in a bed mattress. What this means is when you lie down in bed, excuse me. When you lie down in bed, your mattress will have a bit of give. It will sort of morph and change its shape slightly. But then when you jump up, it'll spring back into shape. And this is what we see with our red blood cells. When our red blood cells are moving around, through our blood vessels, through our capillaries, things can get a bit tight. It needs to sort of be able to bend and squish and move around a little bit, and be quite flexible. Because if it gets jammed, we don't want sort of that occlusion in our blood vessels. And what this does is that it acts like springs. It sort of means that the red blood cells can sort of squish in and then sort of pop back out again and continue on their way. Now, the last one I want to talk about is ankyrin. Now again, keeping it nice and simple, ankyrin it's an anchor. It essentially holds those spectrin fibers together. And it helps them to adhere to the erythrocyte cell membrane. Of course, there's no real point in having these sort of these springs to reinforce and hold the membrane together if we can't adhere it to the cell. If it's just going to fall off, then it's kind of pointless. So what this allows us to do is hold it in place and sort of support the spectrin, these sort of spectrin fibers here, and let them do their job. And that's what we can see here. This is sort of just a schematic of the plasma membrane within a red blood cell. What we can see here is that we've got these spectrin dimers. So these the spectrin protein here. That's then being adhered and attaching to these ankyrin molecules. I was circling actin for some weird reason. And it sort of just, again, adheres it and it holds it in place and, further reinforces it. And that's it, guys. That ends our lecture on biological membranes. So to recap, what did we do today? We looked at the fluid mosaic model. We looked at that fluidity of the plasma membrane. We looked at factors that can change the fluidity. We also looked at factors in which our body can utilize to control the fluidity of plasma membranes, and that is to change the lipid composition to make them either longer or shorter or more or less saturated. Oh, Jack, what's up, man? I've just seen you. You have questions? Yeah, hit me up, man. About the assignment. Sure. Let me go through the learning objectives, and then I'll answer your question. If you want to just post them up, and then I'll address it. Yeah, man. No sweat. No problems at all. Sorry about all the coughing, guys. So then we've looked at and understood the various methods in which lipids can move in that lipid bilayer, so they can move side to side. Again, picturing like a person in a crowd. They can be jostled around, they can move forward, they can move backwards. But of course, you can't sort of very easily turn around and do a handstand in a really crowded area. It sort of needs the assistance of enzymes to be able to do that, because it's just so thermodynamically unstable for it. We've looked at membranous proteins. We've looked at we have discussed the three types. We will, however, be primarily focusing on two: integral and peripheral proteins. And we've looked at some of the differences between them. So with our peripheral proteins, how they line the outside or inside of a cell, that cell membrane, whereas our integral proteins will sort of go straight through that lipid bilayer. And of course, to be able to read and interpret these hydropathy plots and be able to determine how many alpha helical structures that integral protein is composed of, and sort of how many times it would move and pierce through that plasma membrane. Okay, so that pretty much finishes up lecture 11. Which is very exciting, guys. The end is nearly here. We have one more lecture to go, and that's looking at movement of substances through a lipid bilayer, so looking at transport of various substances. However, yeah, if we do have questions about the assignment, I'm more than happy to help you guys out with that, because it is due this Friday. Okay, so the question is there a Turnitin for the assignment that's meant to be up, or are we just printing it off and handing it in tomorrow? There's no Turnitin link. You just simply print it out and hand it in to me tomorrow. If you're worried that you might have printer issues or won't be able to print it and hand it in on time, I always recommend emailing me a copy as well. If anything, I would recommend that you do both. Both send me an email with your assignment attached and print it out and bring it to class. The reason for that is you've got a fail-safe. That way you can sort of say with absolute certainty you have handed in your assignment on time. Because remember, if it's late, you will incur penalties. Now, in regards to the plagiarism sort of aspect of the assignment, I don't rely on Turnitin. I don't have much experience with it. That and it's alarmingly straightforward to see plagiarism in these types of reports. And again, I wouldn't advise even remotely going near that, because otherwise you will have basically academic misconduct, and severe penalties can be thrown down from that, anywhere from receiving a 0 on the assignment, failing the course, or even exclusion from the university in some cases. So my big underlying advice is just don't do it. And again, even if it is referenced, so even if it's referenced in text referencing and referencing in the bibliography, if it's a word-for-word copy-paste that literally isn't in quotation marks, it's still plagiarism. And even then, guys, quotations, not that used that much in science. It very, very rarely. So you should not be sort of directly quoting these sources. You should be paraphrasing, sort of explaining it in your own words, but saying you got the information from those particular sources. So Jack, hope that answers your question, mate. Do you have any more for me? Oh dear. But yeah, also don't forget to make sure that you in-text reference all of your information. Again, your showing where you got that information from. And it's not only the whole, oh, I got the information from here, and you know, acknowledging the people who did that research and source that information. But it's also important in scientific reports, because it helps validate your work. So I could be reading through, say, one of your reports, and you say, you mention a particular piece of information. I go, hang on, that sounds weird. That doesn't sound right. So then I can go, well, I want to follow up on that. Where did you get that information? Was this a dodgy study? Have they just made a revolutionary breakthrough that goes against what, plenty of other people have said? things like that, and that gives me an avenue to find that information. And it also means if you cite it from a source, again under the assumption that it is a peer reviewed and well sourced source of information, you can say, look, I got it from here, and this is what they did. So it's sort of like passing the buck a little bit. But yeah, if there's no other questions, I will probably wrap up this part of the stream here. Let me have a look-see here. Ah, I'm muted. Ah, disaster. Oh, goodness. God, what was I saying? I was saying, oh, that's right. So we'll continue on in class on Friday, finish up the lecture material. It shouldn't take very long. And then what I'm hoping to do is be able to wrap up the workshop stuff from last week and this week and next week, because they are all incredibly similar, and do sort of like a really big joined tutorial thing in which we sort of look at sort of membranes and lipids all in one big hit, because as I said, they are so closely linked. Another question. How much working out do we need to show, if any, for calculating the reciprocals and Vmax? Look, calculate looking at your the inverse stuff. I don't really care. That's I don't need to see that. I obviously do need to see your graph. I'll need to see your like the line of best fit and the linear equation, and show me how you got those numbers. If you just sort of show me your graph and then just put here is my graph, therefore Km is this, Vmax is this, you will lose marks. You're not going to lose all the marks. You may lose some marks, mainly because you need to show me that communication. How did you get those values? So I hope that answers your question with the graphing question. Again, I don't need huge amounts of working out, just the linear equation. Therefore, x-intercept equals blah, and that's going to be your negative km, and y-intercept equals blah, and that's going to be your Vmax. And just compare the inhibited versus non-inhibited enzyme reactions. Awesome, man. Again, if you have any more questions, either hit me up now before we wrap it up, or please send me an email, let me know. I'm happy to help you guys out. Other than that, guys, keep up the hard work. We're nearly at the end, guys. We have one more week of content, then revision. And again, please keep in mind that your end of trimester exam covers everything from week 1 to week 12. But again, I'm going to do a big revision class in week 12 after your 3rd course quiz. So remember, on Friday, you have your 2nd take-home assignment. That is due, so that's due at 12:40. So 10 minutes after class starts. If you do not have your assignment handed in, you will lose 10% for every 24 hours that it is late. Then after that, the Friday of week 12, so our very last day together, you will have your 3rd and final course quiz. Now, may seem a bit harsh that it's so close to the end of trimester exam, but keeping in mind too that everything that is examined in that quiz will also be examined on that end of trimester exam. So you can also think of it as a two bird, one stone kind of a deal. Then what I'm planning to do is a massive revision from week 1 to week 10, go through the slides in which I've basically just taken out all the key things where I would advise you guys focus on that, learn on that. I'll drop some sweet hints, answer some questions. So again, the more prepared you guys are for week 12 and final class, the more you guys will be able to get out of it, the more questions you can ask me with respect to your last exam. Other than that, guys, study hard, don't give up on me now, okay? Remember, uni is a marathon, it's not a sprint, and the finishing line is literally in sight. There's no time for stopping and falling over now, okay? Keep on trucking, cross that finish line, do your best. And on that note, I will see you guys on Friday. See ya.